Thanks very much. So as Philip said, I'll be the first one to give you an RFI presentation if you like. So please listen to the idea, listen to the science, but hopefully this will also introduce the scene for the rest of the RFI presentations that are coming through. And just as a reminder, earlier this year a call went out to all of the participating cohorts to come up with some ideas for these kind of case study or pilot projects as we might call them. Which have then been reviewed and are now trying to be refined. So I'll finish, or towards the end, I'll post some questions that I hope we can touch on further in the breakout sessions. And one of the ideas, certainly for the Team B scientific strategy breakout session, is to work out how we refine and improve the ideas you'll see presented here. So the aims of this project are really designed to fit, of course, within the IHCC. So really, how do we harness the power of having multiple large cohorts? And one aspect of requiring large cohorts that we thought might be relevant is about studying what we've called here uncommon conditions. And I'll expand on that later. Also, I think compared to some of the other project proposals that were put forward, including some of the other ones we put forward, this is what I would see as a kind of opportunity for a quick win project for the IHCC. So this might be something where we can, with relatively little cost and effort using existing data, start to produce a series of manuscripts relatively quickly that help establish the feasibility, the utility of the consortium, as well as establishing it with a brand. So the group I work in, the Cardiovascular Epidemiology Unit, has kind of built its background over the last 10, 15 years on bringing together data from around the world and things like the Emerging Risk Factors Collaboration shown here. This is a paper published 10 years ago now by Emanuele over here and many other colleagues, looking at what happens when you bring together cohort data. So this was 68 cohorts, more than 300,000 participants, and this gave us nearly 13,000 incident cases of coronary heart disease. And we could answer some pretty fundamental questions here. So for example, what's the relationship between various major lipid fractions and risk of disease, adjusting in various ways for different confounding or mediating factors. And one could do that because the commonness of the condition meant that even in a very large number of somewhat smaller cohorts, we could get pretty robust answers. However, when you look in those cohorts at less common conditions, so for example, if we're more interested in something like hemorrhagic stroke, bleeding in the brain, this is just pulling out from one of the supplementary tables a list of those kind of cohorts, which are largely in the thousands or maybe the tens of thousands, but the number of incident hemorrhagic stroke events is very small, often single digits, maybe a few tens, too small to reliably estimate effects, even with very commonly measured risk factors. So we were unable to use that existing framework to start to answer questions about less common conditions, largely because of the size of the original cohorts and the length of follow-up, etc. So how can we get around that with modern epidemiology, 21st century biobank type projects? These are emerging unpublished data that we see as a kind of pilot project for the IHCC in some ways. These were done by a PhD student in my group, Liam Gaziano, who works between the UK and the US in trying to work out how can we harness the power of major biobanks. Here's an example of excellent UK-EU collaboration, which we'll hopefully maintain despite the political situation. This is pulling together two of the cohorts within IHCC, and we intend to add to this, but this is comparing UK Biobank, a more recently recruited general population cohort in the UK with the so-called EPIC cohort, or the cardiovascular component of that. That's a 10-country study, again about 500,000 people recruited across 10 different countries during the 1990s. Now, despite their different recruitment methods and their different outcome phenotyping methods, when we took a very simple ICD code-based approach to identify two of the types of hemorrhagic stroke, either intracerebral or subarachnoid hemorrhage, we could identify several hundred incident cases of each of these events. What you see here with the EPIC data in red and the UK Biobank data in blue is some striking consistency despite these differences, despite, if you like, the crude phenotyping using just a single ICD code. One can get very strikingly similar results from cohorts at different times, but also one can start to dig into if you like, what seems like a major phenotype like hemorrhagic stroke and start to disentangle different subtypes of this. Now, we couldn't have done this using the Emerging Risk Factors Collaboration because we didn't have the scale of data, but by starting with two monumental cohorts, 500,000, with sufficient follow-up time for a number of cases to emerge, one can at least start to understand the risk factors for these diseases, which to date have not been so well understood. There's a lot of interest within the consortium, as has been touched already, in genetic analyses as well. And we said, well, how can we extend this? Often for genetic analyses, we require even greater power than for the classical risk factors I showed you on the previous slide. And so before, or in parallel with the development of the International 100,000 Cohorts Consortium, we tried to see what would we gain by adding other biobanks from around the world, some of which are included in the IHCC. So far, we have participation from 10 cohorts, which should amass something like 4,000 cases. This is just looking at subarachnoid hemorrhage specifically. And from an interim analysis from 7 of these cohorts, with just 2,500 cases, one can already see from the Manhattan plot, the genome-wide association data. This is using existing genome-wide data and ICD-based phenotypes. Again, in these cohorts, one can already see clear genome-wide signal emerging, as well as potentially other signals that we hope to push to genome-wide significance by including additional cohorts. And again, if one looks at this top convincing genome-wide significant signal across these different cohorts, as I said, many of which are in the International 100,000 Cohorts Consortium, one can again see reasonably consistent effects appearing across these cohorts. Now, importantly, any one of these cohorts on its own would not have had sufficient power to detect this signal from the noise when one tests many millions of genetic variants across the genome. And one of the key questions that perhaps we have to grapple with is what are the kinds of questions that one might be able to answer in the IHCC by pooling together data that perhaps go beyond what one could do in any single cohort? And how do we motivate studies to contribute results? I should say the way this has been done is, if you like, in the sort of old-fashioned 20th century way of doing things. This is one PhD student emailing many cohorts. The first challenge being, how do we find out who can even participate in this? Who has enough cases? Who has the right kind of genetic data already? Who might have the right type of ICD code-based phenotyping that we could use to do this? And then if so, sending out analysis plans, getting each center to run the analyses within their own cohort and share the results. Now, I hope, and we've just heard about the ambitions of Team Air, I hope that as part of the International 100,000 Cohorts Consortium, we can improve on that. We can do better. We can make these kind of projects much more efficient, much more streamlined, able to be done. But hopefully I've shown you, at least in these 2 slides, the kind of things that I think we could do within IHCC, and how I think using existing data without generating further data, one can immediately start to perhaps understand the new etiology of less well-studied conditions by virtue of having these very, very large biobank studies. And of course, part of the reason to be here as well is to collaborate. So cohorts that we haven't reached out to would be very welcome to join. And I hope people who feel like they might have the right kind of data in their cohorts are already sitting there and will be interested to join this initial pilot project. Please reach out. So the next few slides are part of the template that was given to people presenting their RFI proposals. So I think cross-cohort needs, well, hopefully I've demonstrated already why this kind of project that we're proposing here requires multiple large cohorts. Simply put, a single cohort just doesn't have the resolution, the power to do it. I feel, as I mentioned, that there's really added value here as well, because the individual cohorts won't be able to answer these questions themselves, that this is really something that we're enabling here that wouldn't otherwise be enabled. And of course, this is trying to pick, if you like, trying to start easy, pick the low-hanging fruit. So really, all that's required here is that we have ICD-coded phenotyping to enable us to get some degree of harmonization or to get around this interoperability problem, to have sufficient cases of the particular conditions we want to look at, and then some kind of either genetic data to contribute to understand the genetic basis of these diseases, or perhaps, if you like, traditional classical risk factor information to look at studying the non-genetic risk factors. So what did we propose as our kind of initial objectives? Well, we said maybe over the next 2 years, we'd try and extend the work that we're doing here on subarachnoid hemorrhage and look at a further 3 uncommon conditions and try and explore the genetic and non-genetic risk factors of both. And we try and do this, as I say, in the kind of traditional way. So this would be asking cohorts to run analyses locally and share results, getting around the initial problems, the policy challenges of whether or not cohorts can begin to share individual level data, and where would we do these analyses. One of the challenges is, how do I find, if you like, just the right kind of outcomes to study? Something that's too common, if you like. Things like coronary heart disease, as I showed you, can easily be studied with many smaller cohorts or studied in individual cohorts. Alternatively, if we go too rare, if we start to look at kind of rare syndromic monogenic conditions, firstly, we won't have enough individuals within each of the even very large cohorts, but also we start to stray into the areas where maybe the etiology isn't best identified through these association studies. So we initially proposed to look at some other cardiometabolic outcomes, given the main focus of our unit. So things like thoracic aortic aneurysm. I've kind of tried to benchmark these just by pulling out some basic stats about the number of events that arise in the US every year of these, to kind of get an idea of how many events we might accrue in the relevant cohorts. So I think there's some thinking to be done here. Particularly, one of the main reviewers' comments we got on our proposal was, well, perhaps we could have a more substantial impact. Perhaps we could show better the capabilities of the IHCC by thinking about non-cardiovascular outcomes as well. So a question to those of you in the room, in your own areas, what might fit into this just right in terms of incident conditions that would be right for study in this kind of consortium. I don't want to get too labored into the detail here, but just to say, as I mentioned, I think this could be done pretty quickly and cheaply. So might need a couple of postdocs. Perhaps the important one here is the second bullet point. How do we motivate people? We live in a data-rich world. That's why we're all here. We all have these enormous cohorts with huge amounts of data. We have many questions we could each answer. How do we motivate cohorts to be able to do the analyses that would be required, especially in these early days where we don't have a framework for data sharing? One solution to that is covering cost, saying, well, actually, for participating cohorts, we'll try and offset the cost of finding a student or a postdoc to do these analyses. But I think that's a challenge that we need to address as a group. I think this is, as we say, things we can do in terms of quick wins. So we hope that there are some clear milestones and timelines here, so I don't propose to go through them all, but merely to say that, first of all, we need to think, as we did, using the subarachnoid hemorrhage template, about what is the phenotype we want to study here, how can we make sure it's a phenotype that can be well explored across different cohorts, and that may take some iteration, of course. How do we ensure we have the right statistical plans that we can then send out to the cohorts as well, that's in an easily understandable way and will fit with different kinds of data from different parts of the world. But then much of the work can be done centrally once the results are shared. So finally, to finish with some challenges. As we said, we saw snapshots of spreadsheets there with cohorts from around the world, but how do we really get to the stage of having a catalog where we know what the cohorts are, we know which ones have the relevant data, we already see who has genotypic data that's imputed, that's available to study, that has the right kinds of outcomes. And ideally, one would want a system where we can already query the whole IHCC to say which cohorts would have enough of the kind of outcomes that makes it worth asking them to participate, and cut down that time substantially. The second is about obtaining relevant permissions. Of course, each of your cohorts probably has different governance structure, and as we've learned, have different paperwork, etc. How do we overcome those hurdles to make it streamlined and efficient? And as I touched on before, how do we motivate cohorts to say, yes, we want to be part of this, but we're going to commit somebody to spend some time analyzing the data, even if it's not the question that's my highest priority. And hopefully some of the successful Team A projects and the Team C policy work will help us to overcome some of those challenges so that we can prosecute these much more efficiently in future. And finally, just to acknowledge the funders who've made these kind of things possible, and Liam, who's done all the hard work here, who's funded by a Rotary group, but also the UK funders who, UK funders and European funders who helped us to build some of the data sets and fund the people who've done some of this work. So thank you very much.
Great, thank you, Adam. We've got time maybe for one question. Yeah, Jeff?
Thanks, Adam. Jeff Ginsburg. So you made it sound so easy to do the subarachnoid hemorrhage study. Could you comment on any of the significant challenges you faced, just even in finding, ICD codes across many different cohorts, or whatever you found? I'd just be curious as to the inherent challenges you saw.
Yeah, sure. Yeah, a number of challenges. So we wrote to a few cohorts who we persuaded it was a good idea and that they should go away and look at how many of this they had. And they came back and said, there's only 10, there's only 20. So that was a big amount of time because typically it wasn't just one email, one person looking up this. The second was about thinking about in detail about that phenotype. So one could take a very crude approach, and we saw very nicely from